Okay, so welcome to the second day of the course. It's good to see all of you back again, awake and fresh for new input into targeted proteomics. And as I already said yesterday you learned already, hopefully, a lot about how to set up a targeted proteomics experiment. You also learned how the data looks like then that the instrument will give you back, and you have also learned how you can identify quite confidently your target peptides then in your data. But what we didn't touch at all was how to quantify, and actually that's the main reason, as we have learned to do targeted proteomics, to get improved quantification. So now we want to dedicate a whole hour just on this topic, how you can use your targeted proteomic data to get quantitative results. And I would like to discuss with you, it's of course a super big topic, quantitative proteomics, but I would like to discuss with you a few most important principles, and in particular look at the differences between relative and absolute quantification. Okay, so I've structured this presentation into 4 parts. In the first part, we will discuss a bit the very fundamental question of why and how mass spectrometry can be quantitative at all. In the second part, we will then look at the principles of relative quantification, followed by absolute quantification. And in the last part, I would like to look at some specific challenges that you face if you want to do absolute protein quantification, and give you some tips and tricks what is important to consider here and to think about in advance. Okay, and again, just as yesterday, whenever there are questions arising from your side, ask them at any time. Good, so how can mass spectrometry be quantitative at all? Or in other words, why is it at all beneficial to generate quantitative data, and why is it often not sufficient just to qualitatively describe which proteins are present in a sample? And one analogy, simplified again, analogy that you can use to highlight these differences between quantitative and qualitative data is the field of photography. So in a way, you can see a qualitative description of a protein sample like a black and white picture of this sample, where every pixel in that picture is only allowed to be either black or white, there or not there, depending on the threshold that you define. And from such a picture, you of course can gain some knowledge. So in this example, you can see that this part of the picture has something to do with greetings from. But many parts of the picture here are rather unclear, and you cannot make a lot of sense out of it. In the moment when you add quantitative information to this picture, and in the world of photography, I say this means when you allow every pixel here to not be only there or not there, black or white, but when you allow it to have different shades of gray. Then this picture changes and becomes much more clear, and you can get the true message of this picture very easily. And exactly the same principle holds true also when you do quantitative proteomics on a protein sample. So if you, for example, want to compare proteins present in the disease cohort and the control cohort, it might well be that you end up with exactly the same protein list. So by just looking at these lists, you cannot differentiate these 2 sample groups. But when you have quantitative information for each of these proteins, it might be that you find some proteins that are higher in the control and other proteins that are lower in the disease. So quantitative data is much more valuable, contains more information than just qualitative data. Pretty obvious. Okay, if this is true, quantitative data is so much better. Why has mass spectrometry or proteomic originally started qualitatively? And why do we only, let's say, in the last 2 decades start to generate quantitative information from our data? And the reason for this is that there's actually a poor correlation between the amount of a peptide in the sample and the signal intensity, the mass spectrometric intensity that we see in a single MS spectrum. So just to make this very clear once more, here's one example. Let's assume we are interested in quantifying this protein here. and we digest this protein with our protease, then in theory all these peptides are equimolar. They have the same concentration. They all come from the same starting amount of protein. However, when we measure these different peptides in mass spectrometry, we will always end up with such a whole range of different peptide intensities. I guess that's what you all observed already, what you all know. The reason why we get these different MS intensities is that the intensity that we measure here for each peptide is not only dependent on the abundance of that peptide. Otherwise, we would expect to be everything here the same. But there are additional factors that also influence which intensity we measure. And these additional factors are, for example, how efficient a peptide is cleaved from the mother protein. This will influence whether it's more intense or low intense. How well do we recover certain peptides during sample preparation? So, for example, very hydrophobic peptides can attach to surfaces, and you lose more of those during your sample prep than of others. Ionization efficiency of peptides. It's just some peptides better ionize, and then you see more of those in your ETHZ MS analysis. Matrix effects. So depending on in which background you are measuring your peptide, it can be that certain matrices enhance ionization and others suppress ionization. So you will get in different backgrounds, different intensities for the same peptides. And it's just, the summary of all of this. Each peptide sequence, each amino acid sequence has its own mass spectrometric detection properties. And the intensity that we see here in the end is really the sum of all those factors that contribute to what we call the specific MS response factor per peptide. And that's the reason why it's super hard to, just by looking at one of these intensities, make a statement about the abundance of this peptide, or finally the abundance of that protein in the sample. Okay? So, but what people have actually figured out pretty fast is that when you focus only onto a single peptide or a single precursor, and you follow the intensity of that single precursor over a chromatographic retention time. So when you measure a peptide many, many times over an HPLC gradient, then you can extract these chromatographic traces that we have also seen yesterday. And the intensity of this chromatographic trace is actually scaling linearly with the abundance of this particular peptide in the sample. And what people usually work with here is either the intensity, the height of this chromatographic trace for quantification, or the area under the curve is the other unit that you can use for quantification. Okay, so this linearity, so this scaling between the concentration of a peptide in the sample and the intensity that you measure in the mass spectrometer is shown here for one example. So this was a peptide that I ordered at the company in a very pure form so that I knew the concentration of that peptide, and then I performed a dilution series from 50 femtomole at the highest concentration down to 50 attomole, I think. And you can see here that you get this nice linear correlation between concentration and intensity, which means that if you double the concentration by a factor of 2, you also double the intensity by a factor of 2. And it's exactly this linear correlation that we use for every quantitative proteomic measurement. That's the underlying assumption why we can do MS1-based quantification, MS2-based quantification. Any type of quantitative proteomic approach assumes that there is this linear dependency between concentration and intensity. That's the basis of all. Now, when you look at this example, you also think, well, that makes a lot of sense, so looks very easy. Of course, that's straightforward. However, there's a problem with this range of linearity, because this range of linearity only holds true in a certain range, in a certain concentration range, as well as in a certain intensity range. Whenever you come above this range, at the higher end, this curve actually flattens out, because at a certain moment you run into problems with detector saturation. That means even though your concentration gets higher, the detector can just not detect more, so the signal will flatten out. At the lower end, you will run into problems with lower limit of detection. So even though you just start measuring background, what is there no longer your analyte, so the curve will flatten out as well. And what's very important, or maybe two more definitions. So the high end of this linear dynamic range is called the upper limit of quantification, and the low end is the lower limit of quantification. So what's very important to understand about this linear dynamic range is that for any type of quantitative proteomics you're doing, it is actually super important that all your measurement points that you collect are coming from this linear dynamic range, because only then the underlying assumption that we have this linear dependency holds true. And this is valid for no matter which type of quantification you do. So this is true for label-free quantification. If both of these data points are coming from a label-free experiment where you have measured the same peptide in 2 different injections. And this also holds true for isotope-labeled quantification. If these 2 data points are a light and a heavy intensity measured in the same injection, it doesn't matter. They all should come from this linear range. In the moment when only one of these 2 points shifts out of this linear range, the ratio that you will obtain, you will get an erroneous quantitative result. Okay? And there is one more complicating thing, and that's the fact that this range of linearity is not something that you can determine for one peptide once and then you know it in your measurement, but it's actually something that is peptide as well as mass spectrometer dependent. So you can have different peptides will have a different range of linearity, and when you move from one instrument to the other, this could change as well. And that's why, in theory, if you want to very accurately determine this range of linearity, what you need to do is you need to have a labeled, pure, not labeled necessary, but you need to have a purified standard molecule from which you can perform a dilution series and then experimentally determine exactly this curve. Now, this is, of course, not what's typically done in all the quantitative proteomics papers that are out there, because to have these purified reference molecules is very expensive and takes a lot of investment up front. So what in most of the papers that are out there, and I'm talking about the label-free relative with thousands of quantitative values that are reported, what the people do is they just assume that all their measurement points are always coming from this linear range. And this assumption is made to give the quantitative values. And of course, it's impossible to quantify thousands of peptides and proteins and perform these linear calibration curves for all of them. However, I just think what's very important is that you are aware of this issue and that this assumption holds true, and that if you want to do very accurate quantification, in theory, you would always first need to check that for your peptides, what's the linear range, and to make sure that all your intensities that you work with are coming from this range. Okay? Good. So if we now want to do our first quantitative proteomic experiment, the situation that you face looks like this, namely that there exists a multitude of different flavors and techniques and applications for quantitative proteomics. And everybody in the field always has the big question for every new project: which of these many different techniques shall I use for my project? What's the best? And of course, it's now not the scope of this presentation to go through all of these different approaches and tell you what's the advantage or the disadvantage. But nevertheless, you can ask yourself a few questions at the beginning of every project, and these questions define in which direction your specific approach should go. For example, it will depend on the type of mass spectrometer or MS platform that you have access to, whether you can do a targeted experiment or shotgun, or what will be the mass spectrometry that you will use. Also, one important question is how precise and accurate do your results have to be? So it can be that for your project, it will be super important to measure small protein changes between samples, and therefore you will need very small coefficient of variance between your replicates, and therefore it will be probably important to do something like isotope-labeled approaches where you can keep your CVs very small. While in other projects, you're mainly interested in large protein changes, and then maybe a CV of 30% is sufficient, and then you could do label-free quantification more easily. Also, the type of sample you're working with is important. Is it tissue, or is it blood, or is it cell cultures? It's more or less easy. Certain labeling approaches can only be done with certain sample types. How large is your project? Do you just want to compare 2 samples extensively or 100 samples? what's your money, what's your budget, how much time can you invest in sample prep and in measurements, and finally, what's the type of quantitative readout that you need. Do you need relative data or do you need absolute data? And while all of those points here are really person and location specific, where you do your measurements, this last point is more general, and I would like to focus for the rest of the presentation on this last point. Okay, just to bring everybody to the same page, what's the definition of relative and absolute quantification? So in relative protein quantification, you always need to compare at least 2 samples. You can have more than 2 samples, but it needs to be at least 2. And the output that you want to generate is always a protein or PTM ratio. So you want to tell at the end of your measurement whether a certain protein or PTM is up or down or not regulated between these 2 or several samples that you have. In absolute protein quantification, the situation is different. You can also do absolute quantification with just a single sample. You cannot do relative quantification with just a single sample. And the output that you want to achieve is always a protein concentration value. So at the end of your measurement, you want to say whether a protein is present in X protein copies per cell or in a certain amount per microgram of tissue extract or per body fluid. All of those are units for absolute protein quantification. So now one important difference between these 2 techniques is that in relative quantification, you always compare exactly the same peptides and the same proteins between the samples. And with absolute data, you can do the same. You can also compare the absolute concentration of the same protein between samples, but you can do one more additional thing. You can also compare the concentration of different proteins within the same sample. And this last point is something that you cannot do with relative, only with absolute data. Is that clear? So if this is the case, why, and we have this nice additional feature that we have with absolute data, why shouldn't we all stop doing relative quantification and just do absolute quantification? But actually this is not a very good idea, because the analytical challenges that you face when you do absolute quantification are much higher than with relative quantification. We will talk about them in the last part of the presentation a bit more clearly. And this is why actually you should always aim for this quantification type that you really need in your measurement. And if it's okay to do relative quantification, you should definitely go for relative quantification and only go through the hassle of doing absolute quantification when it's really needed for your project. Okay, so coming back to this multitude of different approaches. So all of those different approaches can be categorized into techniques that you can use for relative and for absolute quantification. And this categorization is actually very easy, because all of the approaches that you see can be used for relative quantification. You can always do relative quantification with any quantitative mass spectrometric approach. But just a few of the approaches you can also use for absolute protein quantification. So there you need to do something specific. Okay, are there any more questions to why mass spectrometry can be quantitative and how it works with the linear calibration correlation between concentration and MS intensity? All clear? Super. So then let's have a look at some of the principles of relative protein quantification. So all those relative protein quantification approaches that I showed on this overview slide can be further categorized into approaches that work label-free and the approaches that work label-based. And in order to make the difference between the 2 very clear to everybody, I would like to have again a look at a specific example. Let's say this is our target peptide, and we want to measure now these target peptide in 2 samples, control and disease, doing label-free relative quantification with SRM. What do you need to do for this task? Measure this peptide with label-free relative SRM. That's the first thing we need to do. What did you learn yesterday? Yeah, thank you. Rescue me.
Maybe buy a peptide.
Buy a peptide?
That's easiest.
Yeah, but when I buy a peptide, peptide. I mean, you're right, I could buy a non-labeled peptide. So I want to do it label-free here, right? I want to do label-free quantification. So usually that's the way, so when you buy peptides, usually that's the standard approach towards doing isotope-labeled quantification. You would buy then immediate and isotope-labeled peptide. When you stick to label-free quantification, you try to avoid spending money into peptides.
So you need a spectrum, that's where you start.
Mm-hmm.
And the easiest way to get a spectrum is by a peptide.
Mm-hmm. Maybe we can get rescued here. You need an MS2 spectrum because what do you want from this spectrum?
Transitions.
You want the transitions, you want not only transitions.
Intensities, relation. Oh, that's not from MS2 spectrum.
Mm-hmm. But you're right, we want what's called the SRM assay. Before we can measure a peptide, whether it's labeled or isotope labeled, we need an SRM assay, and for this we need an MS2 spectra. So you said one way to get an MS2 spectra is to order a synthetic peptide. What are other ways to get an MS2 spectra? Yeah? I just repeated. Exactly, to build a spectral library. But from what can you build a spectral library? To do a discovery experiment on...
Identify 10,000 peptides or as many as you have in your sample.
So the fastest way is just to use both of these samples and to discovery of them and hope that this peptide pops up.
Yeah.
What if it doesn't pop up in the discovery phase? In this shotgun measurement? What can you do then?
SIM, targeted MSMS.
Do what?
SIM, single eye monitoring in the MS1.
Yeah, would be something you
if
could try as well.
Your sensitivity.
Or you could do a PRM measurement on it or something like that. Mm-hmm. All doesn't work. You never get an MS2 spectra identified for that peptide in any of the techniques you do. What can you do then? Yeah, maybe.
Maybe look in some database repository. If it's there, if it's not there, then you probably need the synthetic peptide.
Actually, this is the very first thing I would do anyway.
First thing is the easiest one.
Yeah, makes you guess how successful your approach was. Exactly. Look in databases. Look in what other people have measured, and if it was never detected before, and you get no empirical data for it, give up and go home. The last thing you could do is then just predict, use these prediction tools, or if you're just talking about a single peptide, I would just measure the doubly charged and the triply charged precursor with a whole Y-ion series or the whole B-ion series, or just measure everything with one peptide. This is no problem at all. And then you can tell if you see it in your SRM measurement or not, for sure. Super, so we need the SRM assay, and we just said already this SRM assay, what it contains. It's this table we have seen many times yesterday with Q1, Q3, collision energy, dwell time, and if we are very experienced and good SRM people, we also have retention time information somehow. Okay, and once we have this assay, we measure both samples, and this is what now we get. So here you see the two chromatograms for the 5 transitions that I see in the control and the disease sample. What do I do now with this data? Yeah?
First of all, you make some replicates.
Oh, very good. I agree. Okay, we have replicates. What do we do now? How do we get the quantitative?
You compare the total intensity of all transitions.
You compare the total intensity of all. So I have many peak areas, right? I have 5 peak areas now. And what would you do with these 5 peak areas?
You sum them up and you compare them, or alternatively you take the ratios of the individual ones and then you see the variation.
Super. These are the 2 main way how people do it. So one option, and that's the option that Skyline does, if you export ratios in Skyline, usually the way Skyline computes a peptide ratio is that it sums up all the intensities in one sample of one channel, sums up all the intensities of the other one, and then it takes the ratio of the 2. If you follow this approach, one nice feature is that depending on the intensity of the peak, you weigh certain transitions more than others. So a more intense transition has a bigger weight on the final ratio weight than small intense ones. And in proteomics this makes sense because often the more intense transitions are the nicer quality ones and the very low ones are lower quality. So by first summing them all up, you put more weight on the more intense ones than on the low intense ones. Another option, what's also done in the field, and there is no right or wrong, or you can do that, but another option is that you first take the ratio of all the individual transition and then make the average of all these ratios, and like this you put the same weight on every transition. Is that clear? And yeah?
Is there a term for the weighted ratio, the first one that you mentioned, like a standardized term that is reported in the literature?
A standardized term?
The weighted ratio of all transitions.
No. I'm pretty well, maybe somebody else has.
Because also it can be non-weighted, right? Or it can be weighted.
I mean, it's just what you do, what's done mathematically by summing transitions first and taking the ratio. This is a weighted output, a weighted, because on the final sum, a high intense one will have contributed more than a low intense intensity. Okay? So it just comes from the nature of summing that you put more weight on some than on others. Okay? But there's no expression, I think, for a specific scientific expression for it. And Skyline, when you work with Skyline, will do this anyway for you. It's just you need to be aware that the value that you export for a peptide ratio will always be the sum of the transitions and then the ratio. Okay? Good. So in this little example, we see that the intensity of the peak is in the control a bit higher than in the disease. So we have a 1.3-fold down regulation, and that would be my result. And I completely agree. So now the next questions that come are, of course, is this significant fold change here or not? And I need replicates to address this. One thing that we also don't address at all here is normalization. So by just looking at this peak and this peak, we cannot judge whether we have loaded the same protein amount from both samples. So it could just be an artifact that here we have loaded a bit less than there. And all these questions are big issues in label-free quantification. And just by looking at this one peak, you cannot judge them at all. So it's very important that you, in label-free, are working reproducible, that you do controls, that you check that you load the same amount, and so on and so forth. Okay, but just to summarize, what are the advantages of these label-free quantification approaches? They are cost and work effective, at least in terms of sample prep. So you do not need to have additional sample labels. You do not need additional steps in your workflow. Usually the sample preparation is pretty straightforward. However, where label-free quantification is not so cost and work effective is in terms of measurement time on the mass spectrometer, because every label-free sample needs to be measured one after another. And there are other approaches where you can first combine samples and then measure them in one measurement, like with TMT or other isobaric labeling strategies. So in terms of measurement time, label-free is actually very expensive. But in terms of sample preparation is very cheap. Label-free quantification is applicable to always all identified or quantified proteins, so you can do large-scale quantitative analysis. You do not increase the sample complexity, so that's a disadvantage when you work with labeling approaches that label your complete proteome. You increase the complexity of your sample by a factor of 2, which is actually a very significant increase and can decrease your sensitivity. And you can apply it to a very large number of samples, so you can compare 100 samples label-free if you want and if you work, if you're careful with all the things that we just discussed already. The disadvantages is that, what we already said, in label-free quantification, you must be very careful that you are reproducible in all the steps of your workflow, in sample preparation, chromatography, mass spectrometric performance. Any variability here will immediately affect your quantitative output. This is the reason why label-free quantification is typically less precise and less accurate than when you work with stable-isotope labeling. Okay. So now let's have a look at all these label-based approaches. And these approaches can be categorized into 4 groups, which differ in the way how isotope labels are introduced into your sample. Oh, yeah, maybe I forgot this. So this is just the facts sheet once more about stable isotope labeling. So what you always do in any of these approaches, you somehow must generate a heavy labeled reference molecule that carries one or several isotope labeled atoms. And the whole theory behind these approaches is that these heavy references are chemically identical to your light, to your endogenous peptides or proteins in the sample. And because of this chemically identical, they behave exactly the same way in terms of sample preparation biases. So if you lose peptides during sample prep, you should lose your light peptide as well as your heavy peptide in the same way. They show the same chromatography, they show the same ionization efficiency, they fragment in the same way. So any variation that happens in these steps, you can account for in the moment when you have a heavy reference molecule present in your sample. And this is why you can measure more precise and more accurate with isotope-labeled references. And the atoms that are typically used in isotope-labeled proteomics are 13C, 15N, or 18O atoms. Deuterium is also used sometimes, but it's a bit less favorable because it can induce chromatographic elution differences. So the elution consistency in chromatography is less valid with deuterium, and this is why people usually try to prefer to work with these atoms. So these are the four groups I already introduced, which differ in the way how isotopes are incorporated in your sample. The first group is via metabolic labeling. So what you do here is you just grow your cells in a media that contains a heavy label, and then all the proteins that are produced in this growth phase will contain the label, and the typical representatives of this type of labeling are, for example, SILEC or 15N labeling. I'm sure you all know about it. The second type of isotope labeling works via chemical reactions. So what you do here is you exploit the reactivity of certain amino acid side chains or of the N or of the C terminus of your peptides and proteins. Typical examples are here demethyl labeling or ICAT, or the two isobaric approaches, TMT or ITRAC. Then we have a third way of introducing the isotope labels. This is, I think, the least favorable way or the least used way, by working with enzymes and by incorporating the isotope in an enzyme reaction. One example here is, for example, 18O labeling. So when you do your tryptic digestion in 18O labeled water, then all your digested peptides will carry at their C terminus 1 or 2 18O labeled atoms. That's also a way to introduce 18O. And the last group of isotope-labeled approaches are the spike-in standards. So here you can work with synthetic peptides, or you can work with protein standards, so completely labeled proteins or completely labeled or C-terminally labeled peptides usually, and you just have them pure or synthetic in hand and you spike them into your sample. And one important difference is, so in those three approaches, you always label a whole proteome. You usually not specific for just certain peptides or proteins. And while here you're peptide or protein specific, you never spike in a whole proteome labeled with these approaches. Yeah?
I had a couple of questions really. So if you go back to the, you don't have to go back, but if you spike, for the label-free quantification, in order to normalize between samples, can't we still spike with a protein, like a known amount of protein, to sort of know how much, is that done or is that not done?
Yeah, that's a very good point. Exactly. So so there are different ways to normalize, and one way, or let's say in general, the advice is to normalize with references, proteins or peptides that came into your sample as early as possible in your workflow, because then you can account for anything that happened from that moment on. So if you want to normalize for sample amount, that you load the same sample amount from all the samples, and you use peptides for that you spiked in just before mass spectrometric measurement, this is not a great thing to do. With peptides that you spike in just before the measurement, you can very nicely normalize for mass spectrometric variation everything that happens afterwards, but nothing before. One way is to work with isotope-labeled proteins, or not necessarily isotope-labeled. If you want to normalize, it's not necessary that they're isotope-labeled. You could just use any purified protein and spike it in. But still, the protein you can only spike in after you have lysed your cells, right? You already needed to do something to your sample usually. Therefore, the very best thing to account for loading variability is to use some proteins that come out of your sample, which you know don't change. This is what's always called housekeeping proteins, and they are just very hard to find. But that would be the perfect control when you know that there are some proteins that don't change, and you monitor those with SRM. This could be very nicely used for normalization. If you want to normalize for total protein amount, one also very good way is to do a shotgun analysis, or to do an analysis where you measure as much proteins as possible and not just a few as with SRM. Because then you can assume that the tick that you see over a lot of data points should be constant overall. This is what people do. And if you do a swath measurement, one great value out of swath measurements is that you can use this tick for normalization of protein loading amount very nicely, even if you then just use a few proteins that you're interested in quantifying. With SRM, usually we're very, very limited in data points that we measure, so to normalize over the tick of SRM measurements is not a good idea at all. There you need to think about something else. Ideally housekeeping proteins. If this doesn't exist, a protein standard that you spike in as early as possible. If you don't have this, a peptide standard, whatever. The earlier you do it, the better you will be able to account for it. Okay?
Just had one more question.
Yeah, go ahead.
You mentioned yesterday that instead of purchasing a labeled peptide, you actually, grew up yeast in 15N label, and
exactly.
then you
That's
mixed it 1:1 ratio with your sample. Now, can you do that with if you have, say, a eukaryotic cell, human cell tissue culture?
Yes, in human tissue culture, there usually the way is not 15N labeling, because it's not, the cells don't grow in these minimal media where you have ammonium sulfate as the only nitrogen source, but you need a lot of other things. But there, Silac, I would say, is the standard way, where you have cells that need certain amino acids that you then add to the media label. So for cell culture or bacteria, to generate these metabolically labeled complete proteomes is usually, let's say, quite straightforward and established. But in the moment when you work with cells that you didn't grow in a media yourself, like if you work with tissue or blood samples from clinics, you cannot do this metabolic labeling. And then people tend to do this chemical labeling or label-free, or you need to think about alternatives. Good. So, again, let's play the same game. This is our target peptide, control disease. Now we want to do label-based relative quantification. What do we need to do now differently compared to this label-free case that we played before? Yeah?
We need to spike a synthetic peptide or isotope-labeled peptide into both samples, and then compare the intensity we get in each sample to this peptide, and then compare them together.
And what about the concentration of this that you spike in?
It should be the same concentration in both. It doesn't matter how much it is.
That's exactly what I wrote. Could be my phrasing. Exactly. That's one way of doing label-based quantification. You take a synthetic peptide and you spike in the same amount in both samples that you're looking at. It doesn't matter so much what's the concentration. It just needs to be the same. Other option is to work with an isotope completely labeled background proteome. It doesn't necessarily need to be a synthetic peptide. But the beauty with the synthetic peptides is that you do not increase the complexity of your sample by just spiking one peptide in, and the beauty is that you are pretty sure about this reference, so when you spike it in an appropriate amount where you can nicely detect it, it just really increases your confidence that you're measuring the right thing. But it does not need to be a highly purified, well concentration controlled peptide if you plan to do relative quantification. Okay. And then we need to measure this peptide, right? And the difference to label-free is that we now do not only need the transition of the light, but we need to add also the transitions of the heavy peptide. Otherwise, we would not get any information for that. Right? So by doing isotope-labeled quantification, our transition list will increase by a factor of 2. So sometimes this can actually be an issue that you then need to split transition lists into 2 runs because you're no longer possible to measure all of them in one go. So here you see now the transitions that I have selected for this peptide. And you can see there are 3 Y ions, and you see the shift that these additional isotopically labeled atoms induce. Usually when you work with tryptic peptides, always this C-terminal lysine residue gets 15N and 13C labeled. It's the standard synthetic peptide that you can purchase from various companies. But what you also see is that I have selected 2 B ions, and these 2 B ions still have exactly the same mass. Is that a mistake, or does that make sense? Did Skyline make a mistake? Yeah?
There is just no C-terminus, right, in these B ions, so it makes sense.
Exactly so, because what I just said, this was a peptide that carried their labeled atoms only at this lysine residue. B ions are coming from the N-terminus, so all the B ions will not carry the heavy label. Okay, so the B ions with such C-terminally labeled peptides will always have exactly the same mass than the B ions of the light peptide. But isn't that a big problem now? We should exclude those transitions then for quantification, no? Because they will pollute. We will see the sum of both forms.
The Q1 is different, so you don't select them together?
Exactly. So the big difference is that we also have this mass difference in Q1. And as the isolation window width in our triple quartz is usually in the range of 1 Dalton, we are here 4 Daltons apart, so it should be no problem. The mass spectrometer should separate it. But you will actually see when you come to swath analysis, where you work with much bigger isolation windows, this can get an issue. So you in SRM, this is completely fine. With swath MS, or when you do not isolate precursors with this 1 Dalton window, you must consider whether you can use B ions or not. Okay, so and then I measure these peptides, and now I get ratios for each sample, because I have the ratio between light and heavy in the control and the ratio in the light and heavy of the disease, and I get 1.3 and 2 fold as the ratio. And as we have spiked in the same amount of the heavy, so these 2 blue curves we know are representing the same peptide in concentration. To take the ratio or to figure out the ratio between the 2 samples, all I need to do is to take the ratio of the ratios. So 2 divided by 1.37, which tells me that my peptide is 1.5-fold upregulated in the disease. You can also see that the difference here is bigger than the difference here. This is how you would compute then from these 2 ratios the final ratio between the 2 samples and get a label-based relative quantitative result, yeah?
The y-axis is very different.
So this is a fake picture. You're absolutely right. It's a very good point, because in theory what you would expect is that the intensity, because they are both spiking the same way, should be similar. So here we have an intensity of 0.8 e to the 6, and here it's 150 e to the 3. And if these would be 2 measurements taken directly after each other on the same instrument, that would be a huge variation. So it would be very bad. Nobody ever got this in any of the presentations so far. Very good. So this is because I just copy-pasted these pictures to make them look nice from some. I'm not even sure this is the same peptide measure. Very good. Okay, but the principle is clear, that you know how to calculate now a label-free ratio, label-free quantitative result and a label-based quantitative result. And here once more the conclusions. So label-based relative quantification is less prone to any variabilities that happen from the moment when your isotope label is added. This is why you're more precise and accurate. And if you work with synthetic peptides, but even when you work with completely labeled proteomes, if you do any of the other 3, labeling approaches, you increase your confidence also because you always look for a peptide that must be present in both channels, right? And this is just, I mean, it's like measuring more transitions for one peptide. It just really also increases the confidence that you're looking at the right thing. We have seen this in this one example yesterday, where when we looked only at the light channel, we couldn't figure out what was the right peptide, but when we added the 15N labeled channel, we increased our confidence what's going on. And this increase in confidence is especially big when you work with synthetic reference peptides, and you're really very sure, unless you don't trust the company that they sent you the right peptide. But the disadvantages are it's work and cost intensive. You cannot apply all methods to all samples. We discussed this already. And depending on the labeling method, you increase your sample complexity significantly with SILAC, with 15N labeling. And I had these cases where in a label-free measurement, I could detect a peptide above the detection limit. But when I went to the SILAC or 15N method, the noise was so big that I was no longer above the threshold. So this can definitely happen. That's all I wanted to say to relative protein quantification. Any more questions, comments? Okay, then let's switch to absolute protein quantification. And as I already said in the beginning, absolute protein quantification you should only perform when you need absolute protein quantification. Yeah?
You still keep referring to protein even though you work on peptides?
Mm-hmm. It's also a very good comment, and you're absolutely right that whenever we want to do protein quantification, we do not need to only combine transition intensities into a final peptide ratio, but we also need to combine peptide ratios into a final protein ratio. And that's actually a bigger issue than combining transition intensities even. And there are various different ways and tools how to do that. I would say the standard way is just to take the average of all peptide ratios that you have measured. That's what often people do, especially in the world of SRM. However, there are also, people have worked a lot on smarter ways, on somehow, again, putting weight on different peptides in a different way, depending on how good their quality is. And I'm happy I can refer here also to Olga Witek's talk. So Olga Witek will do the statistics part tomorrow and the day after, and there this topic will come again back. One easy way to do it in Excel is just to average all peptide all peptide ratios that you obtained to derive a final protein ratio.
What about taking median?
Sure, you can also take the median. I mean, in SRM you rarely have more than 3 peptides anyway, but yeah, you're right, it could make a big difference. I guess this is also there. There is no right or wrong way of doing it, I'm afraid to say. And I'm usually always a fan of putting more data points in, so not to throw out outliers or so, but rather to keep all data points and to somehow combine this information. Taking the median, taking the average are all options what you can do. Even better is to also take into account how reproducible a peptide is, how good it is scored, to combine all of this information into a final ratio as well. And there are software tools out there that were designed to deal with this problem, and you will learn a bit more about them throughout the week.
So maybe I can comment quickly on this. So we will also have a lecture on MS Stats, and there we will also go into how you actually combine those different peptides to get a protein quantification. And this is actually what is currently done, is, at least in MS Stats, is actually median based. So, but we'll get to this.
Super, thanks. Yeah, so MS Stats is this software by the Olga Witek group that you will work with, and there it's one way of, you will learn one way of, very sophisticated way for me to combine peptide ratios and also transition ratios into a final protein value. But you're right, it's really a very important point, and I'm always happy to skip that and just focus on the peptides because it's a big problem and it's often not really addressed in papers or in the field, even though everybody has that problem who deals with proteins. Okay, so which projects are projects where you need absolute protein quantification? Because there are actually not so many out there. Very often relative quantification is absolutely okay to answer your biological question of interest. But there are some approaches where it does make sense to do absolute quantification, and they are all shown here. At least, maybe not all, at least all the ones that I know of. So one typical field of application for absolute protein quantification are protein complex stoichiometries. So in the moment when you want to find out how a certain protein complex assembles, what you need to do is you need to compare the concentration of the different protein components a complex has. So you need to compare different proteins within the same sample. And that's exactly something that you cannot do with relative quantification, but which you can do when you have absolute information on your proteins. The next big field of application is mathematical modeling. So if you want to determine rate constances or kinetic fluxes, in order to solve those mathematical equations, it's often required that you use as an input the absolute concentration value for these equations. Or if you're interested in the energy budget a cell spends on producing a certain protein or a certain protein class, you need to know how many protein copies of that protein are present in the cell. Otherwise, you cannot make these calculations. Then there is the pharmaceutical or biomedical industry with various applications like quality control, food safety, sports drug testing. All of those require that the absolute concentration of a protein in a sample is determined. Same for protein biomarker development. So it's not sufficient to just tell a patient whether he is relatively higher compared to a group control and a disease group, but in the clinic it's really important to measure in a certain plasma or urine sample the absolute concentration of a certain biomarker. And then there is this last field of data cross-comparison. So relative data, unless it was planned to be comparable right from the beginning, can often be not compared retrospectively, which means, for example, if you're interested in comparing the quantitative data of two experiments that were not planned together to be comparable, so not the same reference was used in both relative data sets, it's literally impossible to compare the output. Or if you want to compare data between labs or MS platforms or between two organisms, you can only do these comparisons with relative data if they were measured against the same reference. While absolute data you can always immediately compare to each other. So some cool applications for absolute quantification is to do some new type of comparisons of quantitative data that you maybe even didn't think about when you generated the data initially. Okay. So, and also absolute protein quantification can be categorized in these two different groups of working with isotope labeling and working label-free. Definitely the gold standard in absolute protein quantification is to do label-based quantification. And what you do here is you need to add a labeled reference molecule with a known absolute concentration. That's the only difference compared to this relative quantification we just discussed. The concentration of the reference in absolute quantification needs to be precisely known. And this is typically only achievable with this fourth group that I introduced to you, because only here you have purified reference molecules in your hand from which you can determine the concentration, and then you can spike in a known amount into your sample. So usually 3 different types of molecules are used, either synthetic peptides, often in the literature also referred to as aqua peptides or as stable-isotope-labeled standards. Then there's another type of reference molecule. These are the Q-concats. Does any one of you know what a Q-concat is? Have you heard this expression before? Yeah, maybe one of you can explain it.
All the synthetic peptide that I want are the genome of them are as a plasmid, and then they are produced in the cells, and then have also cleavage sites and are cleaved and released in the sample.
Exactly. So a Q-concat is a concatenation. So one peptide of interest after another is cloned after each other on a plasmid, and like this you create a new artificial protein that never existed before. These peptides can come from completely different proteins, doesn't matter. You can put up to 50 peptides one after another. Like this you create a new protein that you overexpress cell-free or in E. coli in isotope-labeled media. And like this, you get a new isotope-labeled protein that you then spike into your sample of interest. It gets digested together with your analyte and then releases, in theory, 50 equimolar peptides in one go that you can use for quantification. And you can do, in a way, exactly the same as with the Q-concat, also with a complete protein. That's what then the protein standard for absolute quantifications are. And like this, you generate not just peptides, but a whole protein isotope labeled. In a labeled media, you need to purify it, and then when you know the concentration, you can spike the whole protein reference in. Okay, each of these different reference molecules have their specific advantages and disadvantages why you want to work with one or the other. I will just go through it very quickly. So the major advantage of these synthetic peptides is their easy availability. And that's also the reason why most people work with synthetic peptides when it comes to absolute quantification. So there are various companies, Thermo, GPT, I don't want to make any advertisement here, but there are various companies out there where you can just send the sequence, they will produce it for you, and within 4 weeks you usually have these peptides available. Q-concads are also commercially available. If you want to generate your own personal Q-concad, it takes a bit longer until you get them. While protein standards, I mean, there are also some purified proteins that you can buy, of course, but there are just few of them, and typically what people do is they generate their own protein standards, isotope labeled, just in the way I explained to you before.
Just more or less, how much are the synthetic peptides?
In terms of price, all of this is pretty bad. So one aqua peptide that is purified is in the range of 300 euros per peptide. And when we just learned yesterday, you should work with 3 to 5 peptides per protein to get really nice quantitative data. It becomes very expensive to work with these peptides for just a single protein to quantify it. It's over 1,000 euros you need to think about. But also the Q-concads and also the protein standards are expensive and time-consuming to produce. And in general, the price is the big disadvantage of any of those approaches. In terms of multiplexing, so how many proteins can you quantify with these approaches? So most of the SRM projects that report absolute data using aquapeptides, they have maybe 10 to 30 or 50 proteins that they report as absolute values, mainly limited by this factor. The more you get it, the more expensive. So usually people are in that range. With Q-concads, as you can put up to 50 peptides on one plasmid, maybe you can increase this number a bit. And with protein standards, people usually really just work with their protein of interest and just have very few proteins of interest for which they then go through this tedious work of generating an isotope-labeled protein standard. In terms of precision and accuracy, the protein standards are definitely the most precise and most likely, the molecules that most likely produce the most accurate data. And the reason for that is that only with the protein standards you can account for digestion problems. Only when you have a good protein standard you can assume that any unspecific cleavage, not fully full tryptic cleavage, will happen the same to your standard, to your protein, so you can account for these issues. With both of these peptide-centric molecules you cannot account for that. But also with protein standards there are still remaining issues. For example, if you have modifications, PTMs going on your target protein, usually on these standards which are produced cell-free or in another organism, you don't have them very often, so that can be an issue. Or these protein standards are not stable and you do not manage to express them in their really native form. Also, lots of issues can occur with the protein standards, why still these quantitative data that you get are not ideal. But it is the most precise way we have in hand to absolutely quantify. And finally, if you are interested in doing absolute quantification on PTMs, the only direct way for you're left with are working with these synthetic peptides, because only here you can quantitatively, so to 100% introduce certain PTMs by using the appropriate modified amino acid during the synthesis, like phosphorylation or acetylation is pretty easy to introduce. With those molecules, this is much more difficult. Usually what people try to do is they indirectly quantify PTMs by only looking at the non-modified peptide and then say something about the modified version accordingly. If you directly want to look at the modified peptide, you stick more or less to the aquapeptides. Okay, so now we want to do absolute quantification on this target peptide. And we want to do it the most straightforward way, which is called single point absolute quantification. What do you need to do differently to the two examples before? just one thing you need to do.
I mean, I just need a calibration curve for this.
Not
peptide?
even. I even allow you to do only single point
Oh,
calibration.
okay.
You even don't need that.
But it would be good, right?
So you just need to spike a known amount.
You just need to spike a known amount.
You do exactly the same as you have done before for label-based relative quantification. The only difference is that now it doesn't matter what amount you spike in, almost, you need to know this amount precisely. And for that you need to have this purified aqua peptide, which is €300 usually per peptide, or a protein standard or whatever. So let's assume we know we have spiked in 50 femtomole of an isotope-labeled version of this. And then you get, of course, exactly the same measurement points. The ratio hasn't changed. The only difference now is that by knowing the concentration of that blue curve, you can, with single point calibration, immediately calculate the concentration of the red curve simply by saying a 1.37-fold increase in concentration, red versus blue, means 68.5 femtomole per microgram increase in absolute concentration, and the same here. Okay, that's what the only thing you do when you do single point calibration. To do this very easy linear calculation, you need to do two assumptions, though. Maybe you know, the first one I showed already. So there are two underlying assumptions to do this very easy single point calibration. The first one we already mentioned in the beginning. So you assume that your measurement points, so all these intensity values, are actually from within the linear dynamic range of the mass spectrometer. That's the first important assumption. And the second assumption is that this linear correlation that we assume has a slope of 1 and an intercept of 0. And these two assumptions are not always holding true. And therefore, if you want to do even better accurate quantification, you would do what was suggested already. You would do a multiple point calibration curve, which is in a way a dilution series. So you would once more determine what actually is the correlation, the linear correlation of your peptide in the sample. So you would spike your heavy reference not just in one concentration, but in a dilution series in your sample. Your endogenous, of course, stays always constant, and you just use different concentrations of your heavy analyte, and from this you can determine this linear equation, and then you can calculate the concentration of your endogenous peptide. And the big advantage is that by experimentally determine this linear calibration curve, you can account for linear responses that do not have a slope of 1 and that are not going through the axis intercept. However, this always requires multiple injections per sample, so it's not just a single shot and you have a result. You would need to do this calibration curve, and you don't want to do this for every sample that you're interested in. So typically what people do is they generate an external calibration curve once the synthetic aqua peptide arrives. You make this external calibration curve in your sample background, and from this external calibration you can then use the equation, and you can use this in your follow-up measurements on all your samples, and do not need to do many calibration curves in many samples. Okay, yeah?
Is it necessary to make this calibration curve with our background?
When you do it in your background, you first of all learn the true limit of quantification. So the lower limit of quantification will change a lot whether you have a background or whether you have it only in water. There's a second super big problem. When you do it only in water, you will realize that you have a lot of problems with losing peptide due to attachment to surfaces or something. So because you dilute your sample, and the lower the concentration of the sample, the bigger the fraction of peptide that you will lose to whatever. And you will get very strange, I got for some peptides very strange linear correlations when you do them in water. So it's always super important to have other peptides floating around that block things. So I can just recommend you making linear calibration curves in water. Makes no sense for that purpose.
Can you use the same calibration curve for several days?
So what you do when you make this external calibration curve is you usually try to make it again relative to a ratio. So it's not that you use the absolute intensity here on this axis, which of course can change depending on in which shape your mass spectrometer is on a certain day or not. So usually you try to make it relative to something. You plot here ratios that you between a heavy and light version, that you are not prone to this day-to-day variability of mass spectrometers. Yeah?
Is there a reason the signal flattens out towards the top?
It's detector saturation is what people say. But in my experience, it's very hard to flatten out here. It's very easy to flatten out at the bottom. I hardly ever have seen detector saturation on my own. Usually the concentration, how these aqua peptides come, is 5 femtomole per microliter, I think. And even when I need to dilute it at least a little bit, right, with the appropriate buffer. And even there, I'm not reaching the saturation effect. But it might also be LC dependent. Usually the curves that you see are nicely linear, and you ask yourself the question, when do they flatten out? How early? Okay, so, and now just very briefly, I want to mention that there is also an alternative way of doing absolute quantification, not with isotope labeling, but label-free. In the literature, you find papers that describe this absolute label-free based on all the 3 different types of quantification units that we have in proteomics. So there are papers that use spectral counting from shotgun data, or precursor ion intensities from shotgun data, or also fragment ion intensities from targeted data. And in all of those papers, these intensities are then used with various different mathematical models. These are just 5 examples to estimate the absolute abundance from the intensities that you measure here. If you're interested in that, come and see me. I'm happy to explain you more about it. But I guess maybe for the big audience, this is not in interest how this works in detail. But there are various different publications out. And the general difference between the approaches that's, I think, rather easy to understand is that with these isotope-labeled approaches, you always work with 1 reference per analyte. So for every analyte, you have your corresponding reference, while with these absolute label-free estimation approaches, you work with 1 reference calibration curve per proteome. So you no longer have a reference for every peptide or every protein, but you make a calibration curve with a few proteins and use this to transfer concentrations of all peptides and proteins in your sample, which is, of course, more error-prone, but much cheaper as well. So the general advantages of isotope labeling are, as usual, more precise, but expensive and just few targets. The big advantage of absolute label-free is you can do it proteome-wide, so all the peptides that you identify and quantify, you can report an absolute value for in the end, and you really do a large-scale analysis. But the average error that you can expect from these absolute label-free abundance estimations is in the range of 2-fold in average. So this value will be an average 2-fold off from the value that you get over here. And with this error in mind, and by looking back at this picture I showed you in the beginning, I personally think that you must select your absolute quantification approach according to your initial question. So for example, when you're interested in determining protein complex stoichiometry, so when you work in the pharmaceutical or clinical field, often high precision, high accuracy is super important. And then you should use these stable isotope labeling approaches. While when you're in the field of mathematical modeling or these large-scale data cross-comparison, it can become very important to have absolute data from a lot of proteins. And maybe here it's more tolerated to live with this 2-fold error, and you can still make meaningful conclusions. And then also absolute label-free can be a very interesting approach for you to consider. Okay, any more questions to absolute quantification? Great. Then, in the last part, I just quickly would like to draw your attention to a few specific challenges that you face if you plan to do an absolute measurement, and many of them we actually mentioned already. So there are 4 points I want to make. The first one is the optimal peptide selection for absolute quantification. And this we already discussed in detail yesterday. So these were these different requirements that you have if you work with quantitative peptides. And while I already said yesterday, for relative quantification, you maybe can relax on some of them. For accurate absolute quantification, a not fully cleaved peptide from your mother protein will always give an erroneous result. The absolute concentration that you will determine there is just wrong. Every peptide that somehow gets modified will give you a wrong result. So for absolute quantification, you must be much more careful with this selection. And I think this we already discussed a lot. Then there is the second issue, that's complete protein extraction. So for, I think, all the papers that are out that report absolute protein copy numbers per cell, what people usually assume is that they take a cell and they extract 100% of all the proteins in that cell during cell lysis. This is the assumption, I think, usually all the papers that I know behind this value that's reported as protein copies per cell. This is, of course, not reality, because you will always lose proteins either globally, because not every cell was lysed in your cell culture or bacterial culture. And you will also always have local losses, which means you will just lose specific proteins more than others. For example, membrane proteins we know don't get as good in solution as others, or you have some proteins that are just not soluble in your buffer system or something like that. So ideally what you would like to do is you would like to identify these losses, then you can account for them, and then your absolute value that report will be again correct. The problem is that, at least to my knowledge, there is no reliable technique out there that can do that. There exist no tools that tell you how much of the protein fraction was not extracted during your cell lysis protocol. If you find something like that, let me know. I would be very interested in that. So what people do, they try to keep these losses minimal, which means you need to think about the cell lysis, the protein extraction protocol, much more when you do absolute quantification than when you do relative quantification. And what people do here is, for example, they consider to do extraction protocols several times. So I personally worked a lot with yeast and MTB, and we found that when we do extraction with beads beating, it's very, you can extract sometimes factor 3 to 4 more when you do the beads beating in fresh buffer over and over again, and not just once. There is maybe new technologies like the ultra high pressure cycling technology, where you lyse your cells at ultra high pressure, where you can improve this cell lysis. So you can consider trying something like this, or by adding certain improved protein solubilization additives to your buffers, like surfactants, heterotropic reagents, organic solvents. So it's all things that you can try, and that when you want to do absolute quantification makes also a lot of sense to consider and to think about that. And when you do relative quantification, it's maybe less important to find the optimal protocol for your sample prep. Okay, then not only losses can happen at the stage of protein extraction.
Gina?
Yeah?
I think maybe it's important to mention that that basically what we're competing against in the field for standard of absolute quantification is antibody assays, and so some of these there are some problems that apply to antibody assays as well, like some of the ones you just mentioned. There's nothing better about antibody assays for capturing everything that you lose in the cell lysis process. But basically, if you're going to try to do some sort of clinical assay, quantitative assay, absolute quantification, what you're competing against is antibody assays. And actually, when you publish, you'll have people saying, "Oh, did you check that with an antibody assay?" And to make sure you get the same results. And that's something that I guess actually Rudy was co-author on a paper recently, saying we should flip things around and make mass spectrometry the standard by which antibody assays are measured, but that's sort of the competition.
Okay. Good. So not only losses can happen at the stage of protein extraction, but also later during the whole sample process. There are 3 types of losses that I've defined here. So the losses can happen before you do the digestion, when you're still on the level of the protein. For example, once you have extracted your proteins, they still might not be stable in your sample. They can precipitate, they can degrade over time, or they can attach again to the epi surfaces. And the solution to account for those protein losses that happen before digestion is to work with the standard isotope-labeled protein standards, because they can be spiked in after extraction, and then you're assumed to be safe to account for those protein losses. But also after digestion, so once you are on peptide level, you can lose peptides. When you do a C18 cleanup or any other type of cleanup, you will for sure lose peptides. And again, in the moment when you have an isotope-labeled protein standard, or when you now spike in an isotope-labeled peptide standard, that's the way to quite confidently account for these losses. And then there's a 3rd type of loss, which I think often is forgotten, but when you worked a lot with these synthetic standards, as I did, you can actually also lose synthetic standards before you manage to add them into your sample. Actually, it's very easy to lose. And then you think you have added 50 femtomold, but in fact you added only 10. And that is the reason why you must be very careful when you order these synthetic reference peptides, these expensive peptides at high concentration, to make these peptide losses minimal. And well, these peptide losses, they happen also because, as we already said, these peptides can attach to surfaces, and as you work with very purified samples, only your peptide here will attach to the surface and not others as well. So the fraction of what you lose from this aqua peptide is high on surfaces. They can degrade in your solution. You should avoid freezing thawing cycles. So it's known that when you have these peptides and you freeze and thaw them many times, they degrade. They can just degrade when they stand in the autosampler, and you just see that over time the signal gets lower and lower. There are many weird things that can on-go. This is not true for every peptide. There are some peptides who are stable like hell, and you can do anything to them, and you will always measure the same concentration, but others are not. And the tips and tricks that we are giving to the people when you work with these synthetic reference standards is that you should always try to work with low binding vessels and low binding pipetting tips. So you can buy special tips which are designed for keeping losses minimal, and I have personally done a lot of tests with them, and for some peptides there is a 10-fold different whether you pipette the aqua peptide with these special tips or with normal tips. Whenever possible, use glassware. So in general, glassware is less prone to this attachment. Do dilution series in little glass vials. Don't do them in Eppendorf cups. Whenever possible, use a background or a BSA solution to do the blocking of the surfaces. Aliquot your standards upon arrival, and always take a fresh aliquot. Don't do a lot of freezing of thawing cycles to one vial. And I'm a big fan of only purchasing concentrations in solution. So different companies offer either aquapeptides lyophilized or in solution. I personally think when they arrive lyophilized, you need to resolubilize them yourself, you're never sure how much you really manage to get in solution. So I prefer to have companies that ship you the solution, and then you're more sure that the concentration that they determined on that solution is still correct. Yeah. Okay. Yeah. I mean, at this last point, we anyway discussed already that you also need to make sure that you have 100% digestion efficiency in your sample, and again, there are many things you can try. Digestion efficiency is more important in absolute than in relative quantification. Okay, and these are again the take-home message. You can check them out on the slides that you will get. And thanks for your attention, and I'm happy to take more questions.